Hey everyone, welcome to our review of DNA structure and the scientists who discovered it. Let's start by talking about the structure of DNA. We know that it's a double helix, and what that means is that there are two sides and they twist together in this helical shape. But let's break that down a little bit more. So, those two sides of it, or the two backbones of the DNA, are actually made of sugar and phosphate. So, when you see a D in this picture up in the top corner, that's for deoxyribose. That's a type of sugar. And the P is for phosphate. So that's why we call it a sugar phosphate backbone, because you can see sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. And what you can tell about the backbones is that they run in opposite directions of each other. So on this side of the DNA structure, you can see it's 5' going towards 3'. And on this side, the 5' is at the top and the 3' is at the bottom. They run in opposite directions. We call that anti-parallel. But the part about DNA that we usually talk about the most is these nucleotides that are in the middle between the sugar phosphate backbones. And there are four nucleotides: adenine, thymine, guanine, and cytosine. And adenine and thymine always go together, A with T, and guanine and cytosine always go together, G with C. And you can kind of see that it doesn't matter which one comes on the right or on the left, but if it's a G, it's got to be with a C. If it's a T, it's got to be with an A. But overall DNA structure, it's a double helix. It's a twisted staircase type of shape. And there are those sugar phosphate backbones that are twisted around the nucleotide base pairs. And A always goes with T, G always goes with C. Let's talk a little bit more about those scientists who discovered and told us about the structure of DNA. So the two most famous DNA scientists would be Watson and Crick. Watson and Crick actually got together with Wilkins and they published a paper. And that in that paper, they showed off their 3D model of DNA. They built a 3D model actually out of like pieces of metal. And they discovered, or basically showed with their model, that DNA is that double helix shape that we know of, with A bonding with T, G going with C, and that there's a sugar phosphate backbone. They were the ones who really put that together and showed it in a 3D model and published a paper about it. But Watson and Crick could not have done it without the help and information coming from other scientists who they didn't always get along with. So Watson and Crick were on one team at one university, and Wilkins and Franklin were supposed to be working together at another university. Now, Wilkins and Franklin were supposed to be partners, but they didn't get along too well together. They were both working on X ray crystallography, and Rosalind Franklin was excellent at X ray crystallography. She was able to use that X ray crystallography technique to take pictures of DNA and show that it had the double helix shape. She provided the most clear, fantastic pictures of DNA that the other scientists were able to look at and tell that DNA was that double helix shape. That picture that she took with the X ray crystallography machine was called Photo 51. Now, she kind of kept that a secret from the other guys because she knew she was onto something. But Wilkins discovered Photo 51 and he actually shared it with Watson and Crick, which helped them to build their 3D model. So, really, the four of them were the ones who really put it together, though they were not working together as a team. And Watson, Crick, and Franklin and Wilkins were all kind of had bad relations with each other. But Watson, Crick, and Wilkins were awarded a Nobel Prize for their work with DNA. And unfortunately, by that time, Rosalind Franklin had passed away because of the cancer she got due to taking all of the X ray crystallography images. Now, two other scientists who were important in the discovery of DNA were Pauling and Chargaff, but they were a little less directly involved. So, Linus Pauling was a scientist who was in California, and he was working on X-ray crystallography, and he came up with that idea based on his images that DNA is a helix or twisted staircase shape. But he thought that it was going to be a triple helix with three backbones. So he kind of had it wrong. He wasn't able to solve the shape of DNA because he didn't know that it was just going to be a double helix, which is what Rosalind Franklin's images showed. Chargaff is another important scientist in the structure of DNA because he analyzed the chemicals that were within DNA. And he discovered that every time when he took a sample, from different cells, there was always the same amount of adenine as thymine. So he thought, okay, A and T must go together. And there's always the same amount of guanine as cytosine. So C and G must go together. That helped us understand the nucleotide base pairing. So overall, big picture, Pauling says, okay, DNA is a helix shape. Chargaff says A goes with T, G goes with C. Uh, Rosalind Franklin is working on the X-ray crystallography, and she finds a great image of the shape of DNA that shows that it's a double helix. Wilkins, who's supposed to be working with Franklin but really doesn't get along with her, takes that image and shows it to Watson and Crick, who go ahead and use all of the knowledge of all the other scientists to build their 3D model of DNA, which shows that it is a double helix, that it's A with T, G with C, and that there is a sugar phosphate backbone. So overall, all of those scientists' knowledge comes together to give us this information, which we now know about the structure of DNA, that it is the double helix twisted staircase, that A goes with T, G goes with C, and that there's a sugar phosphate backbone. And nowadays we know that that runs in opposite directions. One side goes 3' to 5', the other goes 3' to 5' in the opposite direction. Anyway, thank you for listening.